Hi everybody, this is Christina Howard, and we are going to resume what we did last week, starting with Chapter 2. However, I have to navigate to it first. It turns out when I click Start Presentation, it wants to start at the very first slide, so just let me scroll through these, and there we go. So, this is where we left off with both the Zoom presentation and the last PowerPoint. So we moved from talking about histology techniques, so how the work of histology gets done, and now we're going to dive into the happenings inside of cells, beginning with the cytoplasm of the cells themselves. So let's start. Okay, so like I mentioned both in the Zoom presentation and also last time, a lot of this content is going to be review for a lot of you, but it's important review because it's easy to forget stuff that you're not actively using, and in order for you to be successful on tests, you're going to have to be able to show me that you can link ideas from small to large. So the molecules that make up organelles and link any malfunctions in those molecules to malfunctions of the organelle, and then how that would affect cell and tissue health overall. So it's important to just make sure that we have the nuts and bolts squared away for that reason. So here are sort of the four major life processes. I'm going to use orange this time because reasons. So the four major life processes are just a way to talk about what cells do to stay alive. So I like to refer to these as housekeeping. So these are the things that the cell does, both to support its own life, and there's also elements of whatever the cell's job is in here too. But at the very most basic level, cells have to do certain things to keep running and stay alive. So you can break those tasks down into the following. One is manufacturing, and this is where we get into our macromolecules. So things like nucleic acids, lipid, and most importantly, for most cells, proteins. So proteins include enzymes and structural proteins, and these are the workhorses of the cell. So basically, if you do anything to interrupt protein synthesis, you pretty much kill the cell. Cells can't really live without them. Breakdown is also important. You need to be able to recycle things that are not being used. So this includes lysosomes, vacuoles, and peroxisomes. Lysosomes, vacuoles, and peroxisomes are membrane-enclosed structures that contain breakdown targeted chemicals of some kind. So this can include hydrogen peroxide and hydrolases. Hydrolases are enzymes that use water to perform enzymatic hydrolysis of macromolecules. Of course, we have energy processing. So one of the things that most people take away from high school biology is that the mitochondrion is the powerhouse of the cell. And everybody's like, oh, how dumb is it that I know what that does, but I don't know how to do my taxes? And to that I say, you have a bad attitude about mitochondria, because without them, you would not be alive. And you also would not be able to make any sex steroid hormones. So they're super important. Obviously, I'm a little biased here, but I think mitochondria are dope. Also, they are ancestral endosymbionts, so they didn't always used to be inside cells, which is pretty cool. Chloroplasts do this for plants. Lord, I wish I had chloroplasts. That would be so cool. It would free up a lot of time that I spend eating for me to do other things. But alas, I'm not a plant. And finally, structural support, movement, and communication. So the edge of the cell, the membrane, the cytoskeleton inside of it, and if present, a cell wall. But of course, animal cells don't have cell walls, so we're probably not going to talk about those at all this term. Okay, so this is basically a summary of Chapter 2, so I'm not going to read you this chart, but if you want like sort of the quick and dirty guide to cellular components, this is a good place to look. It's also kind of ready-made flashcard material, so you can put one part of this chart on the back and the other on the front of a card. One note about flashcards I do want to say is that they don't really do you much good if the backside of the flashcard has too much information. So the odds are that if you see this, you're going to be able to rattle off all of this in a flashcard scenario is pretty unlikely. So keep your flashcard sort of contents short and sweet. That's how they'll best serve you. Long form writing is for other things, not flashcards. Okay, so the membranous organelles, my mouth wanted to say membrane enclosed, which is another way to talk about these. So just as the outer cell membrane is a phospholipid bilayer, and I'm going to add phospholipid in here. And just apropos of that, I'm going to be pretty macromolecule forward as I talk about the contents of this stuff, because the chemistry of the macromolecules of life really determine what a cell can do with the components that it has. And so demonstrating mastery or complete understanding of the physiology of the cells we'll be talking about will require that you also understand the significance of the macromolecules that make up the parts of the cell. So that's my rationale for sort of approaching this material this way. Okay, so in order to be a self-contained living thing, you have to have an edge that separates you from the world around you. And so for cells, that's a membrane. A rough endoplasmic reticulum, or the RER, this is, again, made of phospholipids. And actually, so are all of these other things. So these are all just modifications of a bag of water enclosed by a phospholipid bilayer. And what you put in it and what you put in the membrane is going to determine what tasks that little bubble of membrane is capable of carrying out. So the RER is studded with ribosomes, so they sit in the membrane, and it's a site of protein synthesis. It also provides a safe space for newly synthesized proteins to fold correctly, and an apparatus by which proteins that are misfolded can get a chance to refold correctly, which is pretty important. The smooth endoplasmic reticulum is just like the rough, except for instead of synthesizing proteins, it makes lipids and specifically steroids. A steroid is a type of lipid, so these two terms aren't really discrete. One is nested within the other. And then the Golgi apparatus, this is a membranous organelle composed of cisternae. Cisternae is the Latin word for cisterns, plural, which is just a space where you store something. So the Golgi apparatus, or Golgi complex, looks like a stack of pancakes, basically. And things that need to be sorted and packaged go from the cis face of the Golgi, which faces the rough endoplasmic reticulum, towards the trans face and out to the cell's edge. So on the way, things get sorted and packaged and stuck into vesicles or in the membrane of vesicles, and then they get trafficked to wherever it is they need to go. Okay, here's some more membrane enclosed structures. Endosomes. These are just membrane-bound compartments inside of the cell. They come off of the cell membrane, and basically they are so endo means inside and soma means body. So somes mean bodies. These are the result of endocytosis. So let me write that down. And so most endosomes, especially ones that are coated with clathrin, have studded in their membranes little tags that mark the destination where they're supposed to be sent to. Lysosomes, I already mentioned, so these are digestive membrane enclosed organelles, so they are the recycling center of the cells. And any membrane bubble that contains things that are either coming into or going out of the cell, these are called transport vesicles. So here's a link between this concept, and then coated vesicles are associated with clathrin, which is a protein that coats certain vesicles. So these are transport. Mitochondria perform oxidative phosphorylation to make adenosine triphosphate, which is the energy currency of the cell. So I'm going to put some little green shinies around that for cell monies. And then finally, peroxisomes are basically involved in the production of and degradation of hydrogen peroxide, and by virtue of hydrogen peroxide, the degradation of, fatty acids. So if a cell is busy upregulating its performance of fatty acid metabolism, you might expect to see an increase in peroxisomal activity in that cell. Okay, so the plasma membrane is a lipid bilayer, and I want to stress that this little sort of chunk of membrane that you see here, so you can see that there are hydrophilic heads and nonpolar hydrophobic tails, and we've got some cholesterol in here. So cholesterol molecules are structured between the gaps here, equally on both sides of the membrane. So it's not like there's more over here than over here. They're both. So they're gap fillers between fatty acid tails. The lipid bilayer is fluid, meaning that this, like let's say this green fatty acid, this green phospholipid doesn't have to maintain its position in this particular spot. If it wants to like go drift over here, for example, it can. So components of the membrane can move laterally within the membrane freely for the most part, depending on what's in the membrane. So we call it the fluid mosaic model for that reason. And then cholesterol changes the characteristics of the membrane, so they make space between adjacent molecules, changing its fluidity and flexibility. Integral membrane proteins are really, really important. Integral means they are going through and stuck into the membrane, which you can see here. So they include pumps, which move things across the membrane, channels, which do the same thing, but in a different manner, receptors, which receive chemical signals from the outside, linkers, which allow the cell to hook onto stuff that's outside the cell, enzymes, which do a variety of things, depending on what enzyme it is, and structural proteins. These are typically what are called cell adhesion molecules, which we can abbreviate CAMs. And note that these categories aren't mutually exclusive. So for example, move my Epic pen guy over here. So with regard to, apparently I can only use the marker if Epic pen is on. So with regard to the mutual or not mutually exclusive component, nicotinic acetylcholine, we abbreviate that ACH, receptors. These are receptors, but they're also channels by which sodium can pass into the cell. So they're in the middle of the Venn diagram between channels and receptors. That's just one example. I'll give you others as we move along in the term. I just wanted to give you an example so I can prove to you, hey, what I'm telling you is not lies, it's all real. Let's just blow up this. Okay, cool. Okay, so let me get my eraser. I really wish that PowerPoint would just let me do my thing. So I spoke previously about endocytosis, but there's also exocytosis. And I talked about clathrin-encoded vesicles. So phagocytosis, so phagy is to eat, and cytosis means the movement of things into and out of cells. So this phagocytosis is cell eating. And this is where the cell extends little pseudopodia, or pseudopods. Pseudo means pretend, and pod means foot. So they make these little bunny ear type extensions out around whatever the large globule food thing is. And then the tips of the pseudopodia come together, creating a large vesicle, which we call a food vacuole because it contains both the item being consumed, but also a significant amount of water. Penocytosis is kind of like cell taking a little sip of its environment, and along with that sip of fluid, it takes in molecules as well. And then receptor-mediated endocytosis involves molecules that are very specific, interacting with their specific receptor on a patch of membrane, and then once that patch of membrane has accumulated enough bound proteins, that vesicle will form via endocytosis, created a coated vesicle, and the coating on the outside tells the vesicle where it needs to go inside of the cell. Okay, so this should say SEM, not TEM, because remember, TEM is for thin sections. This is an error, so let's cross this out. So endosomes can be viewed as stable cytoplasmic organelles or transient structures. Some are one, some are the other, right? So transient means they appear and then disappear, and then stable means that they're there in the cell always and stick around. So this reveals the presence in the cytoplasm of membrane-enclosed compartments. So there are early endosomes restricted to near the cell membrane. And also from here, many of them are going to return to the plasma membrane as well. Large numbers of vesicles often travel to deeper structures in the cytoplasm, and these are called late endosomes. So early endosomes are really, really close to the cell membrane, and they're called early because they've just formed. Late endosomes have existed for a while, and they are further away from the cell membrane. So you can see a bunch of these all over the place on this picture of a cell. Alrighty, let's go to the next slide. Okay, so we just said that late endosomes often become lysosomes, and these are roughly spherical. Obviously, I'm kind of using spherical loosely, right, because they're not perfect circles, but these function as sites of digestion. So they're super numerous in cells after endocytosis, in part because stuff that cells consume often needs to be degraded. So why not just put some hydrolases inside of the endosome and turn it into a lysosome? Why not? The other source of lysosomes is the Golgi apparatus. So lysosomes don't show up very well on H&E stains, but if you additionally stain with toluidine blue, you can actually see them because toluidine blue likes to react with the stuff that's inside of lysosomes. So here again is a kidney tubule. So just to give you a hint about what you're looking at, this whole thing is the kidney tubule. This is the lumen. Oops, why did I make that into a 4? I must be tired. So this is the lumen. And then here's a cell, here's a cell, here's a cell, here's a cell. So these are the simple cuboidal epithelial cells making up this particular tubule, and you can see a nucleus here too, making this a cell. And then there's cells in this tubule whose nuclei you can't see, but if you were to trace the outline, it would look roughly like that. So that means that all of these little tiny black and purple dots in each cell are lysosomes. You can also see them here in green, so all of these little green circular structures in this cell is going to be the green. And then we've got mitochondria in red among the lysosomes. And just to link what we learned last time with what we learned this time, the blue here is the fluorophore-DAPI. The green fluorophore is probably fluorescein or phalloidin, and the red is rhodamine. So in, these are all proteins. They just happen to be proteins that fluoresce under UV light. Here is TEM, and this is a TEM used correctly because this is a thin slice of the cell. You can see a bunch of big lysosomes all over the place in here. So these are appearing near groups of Golgi cisternae. And you'll notice that they vary in density. So if I erase some of the circles I've drawn here, you can see that a little better. So for example, this one is really, really dense. This one is less electron dense, and that is because this lysosome is actively digesting whatever is inside of it. So it's degrading molecules, meaning that the electron density diminishes somewhat. So you can actually see evidence of that in TEM. And I'm going to pause for a second because Tiki just hurled her stuffed animal off of the top of the cage at me. And I need to go put that back up and put her away. So be okay, I'm back. Sorry about that. My office assistant has the zoomies big time right now. Hopefully she'll tire herself out pretty soon here. Alrighty, so let's move along. So remember when I was talking about the Golgi apparatus, I mentioned the cis face and the trans face of the Golgi, but I didn't provide you with any information about what exactly that means. So this is actually a picture of it, and we call this the endomembrane system. So let me write that down for you. So this involves things being trafficked from the RER, which you can see the cis Golgi faces the RER. You can see polyribosomes translating a bunch of proteins here, and then that protein is probably going to pop into this vesicle and then get sent to the cis Golgi. And then the edges of each stack or cisterna of the Golgi bleb off vesicles, and those vesicles fuse with the next cisterna in the stack, and then those bleb off, and then we get another cisterna, and so on and so forth, until finally they arrive at the trans Golgi and either become a secretory granule or sometimes a lysosome. So remember, I mentioned earlier that in some cases, endosomes do not, or excuse me, in some cases, it's the Golgi that forms endosomes, not the, or Golgi that forms lysosomes, not the endosomes. That's what I meant. So this is kind of the general idea. And as you can see from here, in the direction of the arrows, there is anterograde and retrograde movement. So anterograde and retrograde means forward moving and backward moving. So as far as what you need to know, I am not going to ask you about the specific stuff in these boxes. So for example, do you need to memorize that COPI controls retrograde vesicle movements? No, you do not. You just need sort of the general idea of each step. So the idea that there's a cis and trans face, one's closer to the RER, one's closer to the cell membrane, and then the idea that there's both antero, so cis to trans, and retro, trans to cis, retrograde transport in the Golgi. So that's kind of the idea there. So both taking in things and leaving things. And then over here, which my Epic pen is in the way of, but you have access to this on your PowerPoint that I've provided for you. Lysosomal storage diseases are a group of genetic disorders involving the development of or storage of things inside of lysosomes. These include things like Tay-Sachs disease, which I'll spell for you here. So you can look that up if you're curious about it. But I mentioned, a lot of diseases and pathologies that have really big consequences on quality of life, like Tay-Sachs, often are related to relatively small errors, either in the way a protein's produced or what is supposed to work, or just a seemingly inconsequential error in trafficking of stuff inside the cell. But that spirals out to very large consequences for the organism, including people. Okay, so moving on, let me just trash all this and then we will go to the next slide. Okay, so autophagy. Autophagy means self-eating. So this is basically a process by which cells take apart and recycle parts of themselves that are no longer functioning or that they're not using. So excess or non-functioning organelles or membranes is what happens in autophagy. Those are things are disposed of. So a membrane that appears to emerge from the smooth endoplasmic reticulum basically wraps around the organelles to be destroyed. So again, it puts out those little fake feet, those pseudopodia, and that encloses the organelle to be destroyed, and we call that an autophagosome, which means self-eating body. So I'm going to try and disambiguate the Latin words as much as possible, because the better you are at discerning Latin roots, the better you'll be at scientific terminology. So I'm going to kind of help you with that along the way. So this fuses with a lysosome for digestion of the contents. So the autophagosome by itself isn't good enough to dispose of extra organelles. It needs to fuse with a lysosome. Why? Well, lysosomes contain hydrolases, and hydrolases are what do the work of disposal. So this is a TEM. There are 2 autophagosomes at the upper left. I will circle them for you. Oops, I need to erase this one because I did not include its edge here. And so these are containing portions of rough endoplasmic reticulum. And these are more electron dense than neighboring normal RER. So one of them, the one near the center, may contain mitochondrial membranes. So here we have this one. So these kind of study guys, see those little electron-dense regions that are sort of making little speckles? Those are ribosomes. And then the reason this one near the center might contain both is because this region looks like RER, and here we can see what look like stacks of membrane as you would see in the internal mitochondrial membrane. So we don't know that for sure, but based on their morphology, we can assume that that's probably what they are. And then we also have a residual body, which basically is what's left over once digestion has occurred. Okay, so proteasomes are protein complexes that work independently of lysosomes. So basically, in some cases, they're very specific cases typically, autophagosomes can degenerate what their contents without the help of the lysosome, but in some cases not. So these are just big protein complexes that go to work attacking proteins, which are not themselves organelles, without the involvement of lysosomes. So here we have lysosomes, and in this picture, we stain them with acid phosphatase, not because we're looking at lysosomes specifically, but because we want to be able to see the difference between autophagy that is proteasome mediated, which is occurring here, and autophagy that is lysosome mediated, which is occurring in the sort of black circles. So that's the reason for that differential staining there. Okay, so I showed you the little studs of the rough endoplasmic reticulum in the previous slides. And now we'll look at an electron micrograph, this being a TEM again. How do I know? Well, it's a thin section. So you can see membranes arranged in stacks, and on them you can see all of these little small electron dense regions. Those are the ribosomes. So that's how I can tell. So these happen to be in the chief cell of the stomach. So the chief or zymogenic cells make digestive enzymes, not acid. So they're also called zymogenic cells because they make enzymes. So these are going to make things like pepsinogen, which helps with protein digestion. So it makes sense that they are sort of jam-packed with rough endoplasmic reticulum if their primary purpose is to make a protein for secretion into the stomach lumen. And then you can also see a mitochondrion with its outer and inner mitochondrial membrane there. So polyribosomes are what you're seeing. Those are those electron-dense regions, and those are just groups of ribosomes that participate in translating proteins. So it's really cool and interesting that you can see all of that stuff right in this TEM. So the RER manufactures proteins that are supposed to be secreted, or ones that are destined for other organelles and membranes. But remember, we've got both cytoplasmic ribosomes and ribosomes that are embedded in the RER. And you can make the generalization that stuff that is supposed to be going out of the cell is primarily the job of the RER because of its relationship with the Golgi apparatus. And proteins that are going to stay within the cytoplasm, not necessarily going to another organelle, but just sit in the cytoplasm, those tend to be synthesized by cytoplasmic ribosomes. So that's a generalization that you can safely make. Okay, so the smooth ER lacks ribosomes, and it synthesizes lipids. So it looks like the RER a little bit, and it's actually contiguous with it, so the membranes are not separate. It's just that it doesn't have any ribosomes on it, which is why we call it smooth. So this synthesizes lipids, a variety of them. So you would expect to see lots of smooth endoplasmic reticulum anywhere lipids were produced. So one example is bile is an emulsifier for fat. And so if you have an underdeveloped SER in liver cells in a newborn, you can't convert bilirubin, which is a bile pigment, into a form that can be excreted, and that causes a buildup of bilirubin in the cells and tissues, which gives the infants a yellow appearance. So smooth ER is where lipid synthesis and carbohydrate metabolism is present. So an example of some cells where you'd expect to see a significant amount of smooth ER is any cell that's going to synthesize lipids actively. So one example of that would be cells that make steroid hormones. And these include cells in the adrenal cortex and also cells in gonad, as well as in the liver. So they're all over the place. Okay, so this is just an overview of the Golgi apparatus and vesicular trafficking. So note that there is anterograde and retrograde transport, so both towards the cell's edge or boundary and away from it. And these are going to become endosomes or become part of an exocytosis body, respectively. But this basically just shows you the relationship between the RER and the Golgi apparatus. Mitochondria are abundant in cells that generate and expend large amounts of energy. So most cells have mitochondria. Not all of them do. Some of them used to, but no longer do. So for example, red blood cells don't have mitochondria. And that's because mitochondria use oxygen to produce ATP, and red blood cells are specialized for oxygen delivery, so they can't have mitochondria because then they would be using their oxygen on the way to dropping it off. That would be like if I was an ice cream man and I ate all the ice cream on the way to my delivery destinations. I'd be a pretty crappy ice cream man if I was doing that. So similarly, red blood cells kind of spit out all their mitochondria right before they become mature. So these work because they create a little proton dam between the outer and inner mitochondrial membranes. So if you were to zoom in on this intermembrane space, you would see lots of protons. And that creates a pretty steep gradient such that protons want to flow from here into here. And that causes the ATP synthase to spin around and synthesize ATP. So it converts kinetic energy of spinning into the chemical energy necessary to form covalent bonds. So bonding ADP to an inorganic phosphate forming ATP. So that's kind of a quick and dirty guide to the goings-on in the mitochondrion. All of the other reactions, for example, the matrix reactions like the Krebs cycle, are basically designed to produce reduced electron carriers that powers the process I just described. Okay, peroxisomes. These are going to produce and degrade hydrogen peroxide. So hydrogen peroxide is required to inactivate certain potentially toxic molecules and also break down things like alkaloids, so some prescription drugs. So you have a lot of peroxisomes in the cells of your waste disposal organs. So liver and kidneys are rife with these things. So I want to take a moment to step aside from the content that we're discussing right now and make a point, and that is all over the internet you'll see, like Instagram and Facebook are particularly guilty of this, ads for like detox treatments. So these include like body masks that you wrap around your belly to detox your belly somehow, or drinks that are for detoxing or detoxing by juice cleansing, or detoxing by drinking activated charcoal. You have two very powerful detoxifiers built into you, assuming you have a liver and both kidneys, or even just one kidney. And they do a wonderful job of taking care of the waste in your body all by themselves. Some waste products do come out a little bit in your skin, but their effect is negligible. So similarly, if you've been really going hard at prescription drugs or alcohol, for example, getting in the sauna is not going to help you. It might feel pretty good. I'm not going to lie. Saunas are great, but are they effective detoxifiers? Nope, absolutely not. And also, the only time you should be drinking activated charcoal in large quantities is if you are needing to get your stomach pumped. So activated charcoal's surface area helps to mop up potential poisons before the clinician in the hospital forces them out of you by making you vomit. So just be wary of products that are for quote unquote detox, and be thankful that you have a detoxification system built into you. All that other stuff is just ways to extract money from your wallet by engendering fear and making you feel dirty. It's a marketing tactic. Regard it with suspicion. Okay. So now I'm hopping off my soapbox and we'll talk about peroxisomes again because I think they're neat. So over here we have a cell, and this is an endothelial cell from some organ. We don't know which, doesn't really matter. But the peroxisomes here have been stained green, and then you can see the DAPI stained nucleus here. So all of these little guys are peroxisomes. How do we know? Well, we've designed an antibody labeled for a peroxisome-specific something or other, probably one of the enzymes associated with peroxisomes only. And then we've got mitochondria, which are surrounding. And you'll note that we typically illustrate mitochondria as being kind of a bean shape, and they often are, but not always. Sometimes they can be an interconnecting lattice work of membranes. And this is just an important thing to note, that the scientific illustrations in your textbooks don't always directly reflect reality. Sometimes, mitochondria are membranes. Membranes are fluid, they're stretchy, they can take any shape, and sometimes they don't look like a bean, so just be aware of that. Okay, so non-membranous organelles. So these ones are not made of a phospholipid bilayer, they are made of something else. So microtubules form from elements of the cytoskeleton. So they're made of a protein called tubulin. So this is a specific protein. And then a dimer is a complex of two, oops, I didn't mean to write B, I meant to write a T. Two tubulin proteins together. So if you were to look at them, they would look like one globular tubular protein, and then let me pick another one, and another one. So these glob-like proteins form dimers, and then these dimers spontaneously and very rapidly, I might add, polymerize and depolymerize inside of the cell to form pathways for transport, and also in some cases to move the cell along. So when cells crawl along surfaces, for example, the wandering macrophages that crawl along the inside surface of your lungs, they're extending microtubules at their leading edge, and they are depolymerizing them at their trailing edge. Microtubules are also critical for exoplasmic transport, so the transport of proteins primarily towards or away from the axon of a neuron. Filaments are also part of the cytoskeleton. They're smaller. Actin filaments form from actin, and these are flexible chains. And then intermediate filaments, which are more rope-like, these allow cells to withstand shearing forces, so resistance to being torn apart. Centrioles are short cylinders made of microtubules. So you can use microtubules by themselves, or you can use them to build larger things. And these are basically two little paired cylindrical structures made out of microtubules that are involved both in cell division and organizing cell division. So the two centrioles sort of migrate to opposite ends of the cell, centrosomes migrate to opposite ends of the cell and form the axis for the mitotic spindle, and or give rise to the basal parts of cilia. So cilia are a microtubule-mediated cell production. Ribosomes, I've mentioned previously, so these are a weird complex of ribosomal RNA, so a nucleic acid molecule, and proteins as well, so they're both. And so these are essential for protein synthesis. The work of synthesizing proteins doesn't get done without these things. So you got to have them, and most cells have them both embedded in the rough endoplasmic reticulum and also just floating about in the cytoplasm. Okay, so here's just a picture showing you microtubules. So tubulin dimers are one light blue and one dark blue circle, and then they coil up into this tube shape. Intermediate filaments are more like a really big cable that you would use to stabilize something. And microfilaments are made of actin, and these are, so globular actin is G-actin, and then when it forms a filament, we call it F-actin. And these are flexible and sort of string-like. Critical for muscular contraction, I might add. I'll explain more about that later. Okay, so I promised I would show you along the way some pathologies. So pathology is a place where there's some illness at play, so some aberration from normal homeostasis. And histopathology is the study of disease processes in cells and recognizing them visually using histology techniques. So one place where you can see this is abnormalities in microtubules and microfilaments. So because microtubules and microfilaments both play a role in maintaining the shape of a cell, when they start to go wrong, you can see that on a microscope slide pretty quickly. So Mallory bodies, named after the person who discovered them, Dr. Mallory, are accumulation of keratin intermediate filament. So that's the specific intermediate filament type. And because of the nature of these molecularly, they tend to clump up. And they're frequently associated with very specific cell injuries. So cirrhosis, which most humans acquire by alcoholism, so the chronic overconsumption of alcohol, causes hepatocytes to malfunction in a way that generates Mallory bodies. So the arrows here, and I'll circle these too, are pointing to Mallory bodies in a cirrhotic liver. And this is a pretty bad case. In someone with early stage cirrhosis, you wouldn't see as many of these. But since I also run a cadaver lab and I've seen decedents that have passed partially of cirrhosis, what I can tell you is that when cirrhosis is pretty advanced, because these filaments are fibers, you can really tell the difference between a healthy liver and one that's experiencing Mallory bodies due to cirrhosis. If you cut into the liver of somebody who has alcoholic liver cirrhosis, their liver is stiff and fibrous and difficult to cut through compared with a healthy liver in a decedent, which is not. It's dense because it's fixed in formaldehyde, but it's not fibrous. So the sort of cumulative outcome of a bunch of these is a change in whole organ morphology, which is not bad. Or excuse me, which is very bad, not good. So lymphocytes and macrophages surround cells containing Mallory bodies, and those end up destroying those cells, which obviously a loss of hepatocytes means a loss of liver function. So immune cells recognize these cells as being abnormal and destroy them, and then you lose hepatocytes, which means liver function declines. So, for example, a place where that might matter clinically is if you were suffering from cirrhosis, your doctor might do some tests on levels of enzymes and stuff, which is sort of a way to indirectly assess how many of your hepatocytes are functioning. So if the levels of your liver proteins are low, they might give you a percentage. So you're down to like 60% liver function or something. So they're giving you a percent of normal function for that. So booze is fun. It's nice to relax with some wine and whatever, but go with caution because it is definitely not good for you. Okay, so here's centrioles. Here we have a 120,000 XTEM, so that's some serious mag power. And then a drawing of the same thing. So you can see that these are cylindrical arrays of microtubules with adhesion proteins between them. And these form two short cylinders that are always orthogonal to each other. So orthogonal is a fancy way of saying at right or 90-degree angles to each other. So they're always paired, and they're always at right angles to each other, and they're always next to a dense matrix of free tubulin. So the free tubulin floating around here can rapidly sort of hop on to one or the other ends of the centriole and extend any one of these microtubules in the triplet. So that's what happens, for example, when a cell is getting ready to divide. Okay, inclusions are, and you just saw Mallory bodies, which are inclusions. So an inclusion is a glob of something in the cytoplasm, but it's not surrounded by a membrane. So these are sort of naked aggregations of stuff. Some of them are normal, some of them are not. So, for example, fat droplets are accumulations of lipids, specifically triglycerides in adipocytes. And they can also be aggregations of steroids in the cells of the gonad and of the adrenal cortex. Glycogen granules, these are large branching chains of glucose, so they are a starch-like molecule that is used for carb storage in animal cells. Lipofuscin is a kind of yellowish brown pigment found in stable and non-dividing cells. And then hemosiderin, hemo should suggest to you that this is related to blood. So these are proteins that are denatured, meaning unfolded, and they're bound to iron, specifically ferrous iron. So hemosiderin, by nature of what it is a breakdown product of hemoglobin. So the pigment part pops off, and you get hemosiderin and then a sequence of other sort of waste products, including bilirubin and others. So here we have an electron micrograph. This is a TEM, transmission electron micrograph of a liver cell, and all of these dark things are glycogen inclusions here. Okay, and now we are at chapter 3, so I'm actually going to stop here just so that it's easy for my video to upload, and I will record chapter 3 sometime tomorrow, and I'll lecture on both of them tomorrow evening in the Wednesday evening lecture. And then once I get done with this particular PowerPoint, so the end of chapter 3, that will conclude this week's testable material for your your quiz that will be available starting on Monday. So until next time, and thank you for your attention.